I'm Tiana Sarso.
And I'm Ashna Kumar.
As we all know, cancer treatments aren't the most effective, and the scientific community is always looking for new ways to treat cancer. It is important that we have a way of detecting viral infections because being able to detect them early means that we'll be able to treat patients earlier on. There was a recently published article focusing on protein-based sensors that could possibly detect viral infections or kill cancer cells. This project is driven by a type of protein called the zinc finger, which is a DNA-binding protein that can detect a wide variety of DNA sequences. Here's a video describing how zinc fingers work.
Zinc finger domains are structural motifs used by a large class of DNA-binding proteins. They use centrally coordinated zinc atoms as crucial structural elements. The zinc atom is coordinated by 2 cysteine residues from the beta sheet and 2 histidine residues from the alpha helix. A single zinc finger domain is only large enough to bind a few bases of DNA. Zinc fingers are often found in tandem repeats as part of a larger DNA binding region. The helical region of each zinc finger rests in the major groove of the DNA. Amino acid side chains project out from the helix and contact bases in the DNA. The identities of these side chains determine the precise DNA sequence recognized by each zinc finger. Assembling different zinc finger motifs allows precise control over the sequence specificity of the protein.
Cancer cells are present in the body due to rapid and uncontrolled cell divisions. Non-malignant tumors are masses of cells that divide rapidly. However, cancer cells do not form until a cell from this tumor strays and spreads to another part of the body. This is called metastasis. These cells, once they stray away from the tumor, colonize at different sites in the body, forming new tumors.
A tumor that does this is called a malignant tumor. When cells migrate away from the tumor, they can affect the function of distant organs. Physicians can detect metastasis by looking for specific types of intermediate filament proteins in places where they should not be, such as the bloodstream. Physicians can also examine lymph nodes to indicate if cancer has spread.
Viral infections can also be indicated based on this procedure. According to the Mayo Clinic, viral infections, including AIDS, chickenpox, and the common cold.
Viruses can also cause cancer. Virus is an accumulation of proteins surrounding either a core of RNA or DNA. They can multiply by infecting cells and using the host to synthesize the proteins encoded in their genomes to make more virus copies. Viruses can carry a handful of genes, which facilitate the identification of genes involved in cancer. A specific gene for the virus promotes an uncontrolled cell division. This gene is known as an oncogene, which is a cancer-causing gene. This specific gene, along with other factors, can cause cancer.
Our news correspondent, Emily, has more information about this recent development in the study of protein-based sensors that can possibly detect viral infections and kill cancer cells. She is now live at MIT.
Thanks, Ashna. I'm here at MIT with James Collins, the Professor of Medical Engineering and Science in MIT's Department of Biological Engineering and Institute of Medical Engineering and Science. Hi, James. How are you?
I'm good, thanks.
Tell us more about this recent discovery you and your engineering team have found.
Well, here at MIT, we've developed this modular system of proteins that can detect specific DNA sequences in a cell and then trigger a specific response like cell death. Our project is driven by a type of protein called zinc fingers, which is a DNA binding protein and that can detect a wide variety of DNA sequences.
How did you create this new system?
Well, the zinc finger proteins already have their function, which is to bind to DNA. But what they lack is a consequence to this function. We wanted to make use of their already existing function by adding an additional consequence, that is some sort of fluorescent protein activity.
And once the zinc fingers are linked to the fluorescent protein, how did you determine that a cell is infected with the virus?
We linked green fluorescent proteins, also known as GFP, production to the zinc fingers recognition of a DNA sequence from an adenovirus, so that any cell infected with this virus would glow green.
Aside from using this process to identify infected cells, how else would this process help with someone dealing with an infection?
Because this is just a modular, you can pretty much program it to do anything you want. You can program it to kill itself, or even to send out a signal to the immune system that would eventually cause its death.
Since this process is still in its early phases, what else can we expect from future versions of this system?
In the future, we can design these systems to bind to DNA sequences found in cancerous genes and then produce transcription factors that would activate the cell's own programmed cell death pathways.
Why should the public pay more attention to this developing system, and what important functions will it have as you continue to research it?
Well, this is something that could potentially run our future. This could really change our lives for the better, and it is possible that some of us might see this being applied in the near future.
Thank you for your time.
My pleasure. Thanks for having me.
So now we have another scientist on the line giving her opinion on this process. Back to you, Tiana.
Thank you, Emily. Although James has put in a lot of work, many have stated their opinions objecting to this process because of the generation of genetically modifying organisms. Now we have Amanda Smith from Duke University, who is a professor of medical engineering and science in Duke's Department of Biological Engineering. She is phoning in to give us her opposition to this recent study.
Thanks for having me.
So what is it exactly that you find to be unethical about James' study?
I think it is unethical that scientists are programming these protein groups to genetically modify these patients to improve their health.
What do you think should be changed to make it more ethical?
I think they need to find a better way to understand how to use the protein zinc fingers so they will not harm the organism and or change their genetic makeup. I mainly just think that there needs to be more research done on this so that they understand these proteins more before they really test it to ensure that there is not any harm done to people.
Well, thank you for your opinion, Amanda, and thank you for coming on our show. Now here's Ashna getting to know the public's opinion on this study.
Right now, zinc fingers are in the lab as a modular, but could potentially be used on humans. What is your opinion about testing these zinc fingers on humans not knowing its effects but has the potential to cure a disease?
I think that even though we don't know what the potential risks of it are, the possibility of actually finding a cure and a diagnosis for these unknown diseases is worth the risk.
Thank you.
No, I'm all for curing diseases, but if it harms people in the process, I think we should find a better way.
I think that it's okay, especially if it has the potential of curing a disease like cancer.
That's all we have for today, and tune in next week for another edition of Nucleic News.